Picture two skeletons side by side in an ancient burial site somewhere in Central Europe. The date is approximately 4,500 years ago. The late Neolithic, early Bronze Age transition, a period when the world was in the middle of one of the most significant social reorganizations in human history. A team of archaeologists excavates them carefully. Both individuals appear to have been buried together deliberately, their positioning suggesting some kind of intentional placement. Analysis of the skeletal remains begins the process of reconstructing who these people were. Their sex, their age at death, their health history encoded in bone chemistry and wear patterns, their diet encoded in stable isotopes, their geographic origins encoded in strontium ratios from the local geology versus the geology of wherever they grew up. And then ancient DNA analysis enters the picture, extracting genetic material from teeth and comparing it to other individuals from the same site and from sites across the region. The results, when they come back, reveal something about the relationship between these two individuals and about the reproductive patterns of the broader population they belonged to that sits at the intersection of biology, anthropology, archaeology, and one of the most politically charged questions in all of human behavioral science. The question of how many partners the average ancient human had is one of those questions that sounds simple, possibly salacious, and turns out to be one of the most revealing windows into the entire structure of ancient human social life available to modern researchers. Because the answer, which is neither as wild as sensational headlines suggest, nor as simple as any single modern moral framework would prefer, reflects the intersection of evolutionary biology, ecological constraints, social organization, and the specific historical circumstances of different periods and populations in ways that require actual examination rather than assumption. Here is what the evidence actually shows across multiple lines of inquiry about the mating patterns of ancient humans. Begin with the biological evidence, because the body carries a surprising amount of information about the mating system of a species in its anatomy, and the human body is more informative on this question than most people realize. Comparative primatology provides the baseline framework. Among the great apes and other primates, the relationship between mating system and physical anatomy is consistent enough to be predictive. Species with high levels of male-male reproductive competition, where many males compete for access to a small number of females who do most of the reproducing, show dramatic sexual size dimorphism, with males substantially larger than females because larger males win more competitive encounters and therefore have higher reproductive success. Gorillas show extreme sexual size dimorphism, with males roughly twice the body mass of females, consistent with a polygynous mating system where a single dominant silverback monopolizes reproductive access to a harem of females. Species with lower male-male reproductive competition, where reproductive success is distributed more evenly across males, show reduced sexual size dimorphism because the selective advantage of large male body size is weaker. Gibbons, which are largely monogamous, show minimal sexual size dimorphism. Chimpanzees, which have a promiscuous mating system where multiple males mate with each receptive female, show intermediate dimorphism. Humans show modest sexual size dimorphism. Males average approximately 15 to 20% larger than females in body mass, with variation across populations. This places humans in a specific position on the primate mating system spectrum, clearly above the minimal dimorphism of monogamous species, clearly below the extreme dimorphism of polygynous harem-forming species like gorillas. The anatomical evidence suggests a mating system with some degree of male-male reproductive competition, but less extreme competition than highly polygynous species. Testis size provides an independent and complementary line of anatomical evidence. In species with high levels of female promiscuity, where a female mates with multiple males whose sperm then compete within the female reproductive tract for fertilization, males evolve larger testes relative to body size because sperm competition favors males who can deliver more sperm into the competitive environment. Chimpanzees, whose mating system involves multiple males mating with each receptive female, have very large testes relative to body size. Gorillas, where a single dominant male has near-exclusive access to females and sperm competition is minimal, have very small testes relative to body size. Human testis size falls between these extremes, larger than gorillas, smaller than chimpanzees. This intermediate testis size is consistent with moderate levels of historical female promiscuity. Not the extreme multi-male mating of chimpanzees, but more than the near-exclusive male access that gorilla mating structure involves. The anatomy suggests a mating system somewhere between pair-bonded monogamy and high promiscuity, a pattern consistent with what anthropologists describe as mild polygyny combined with some degree of extra-pair mating. The ancient DNA revolution has transformed the study of ancient mating patterns in ways that earlier generations of researchers could only approximate through theoretical modeling. By extracting and comparing genetic material from individuals buried at the same sites across multiple generations, researchers can now directly identify parent-offspring relationships, reconstruct family structures, and measure the genetic signatures of different mating systems in past populations. One of the most informative genetic signatures of historical mating patterns is the ratio of genetic diversity on the Y chromosome compared to the mitochondrial genome. The Y chromosome is transmitted exclusively from father to son, while the mitochondrial genome is transmitted from mother to all children. If a mating system involves high male reproductive skew, where a small number of men father most of the children while many men reproduce little or not at all, the effective population size of Y chromosome lineages will be reduced relative to mitochondrial lineages. Reduced Y chromosome diversity relative to mitochondrial diversity is a genetic signature of polygyny. Analysis of ancient and modern human genetic diversity has found exactly this signature in certain time periods and regions. Research published in the mid-2000s identified a dramatic reduction in Y chromosome diversity approximately 5 to 7,000 years ago, specifically in the period following the spread of pastoralist populations across Eurasia and the expansion of early agricultural civilizations. This bottleneck in Y chromosome diversity, affecting populations across multiple continents during the Bronze Age, is consistent with a period of dramatically increased male reproductive skew, a period when a small number of males were fathering dramatically disproportionate shares of the next generation. The magnitude of this Bronze Age Y chromosome bottleneck is striking. The genetic data suggest that during this period, perhaps one man was fathering children for every 17 women of reproductive age in affected populations, a degree of male reproductive skew far exceeding anything observed in modern populations and suggesting a brief but significant historical period of extreme polygyny or reproductive monopolization by elite males. The same ancient DNA analyses that identified this bottleneck have helped clarify what came before and after it. Before the Bronze Age transition, in Paleolithic and early Neolithic periods, the ratio of Y chromosome to mitochondrial diversity is closer to what would be expected under more equitable male reproductive participation. After the Bronze Age bottleneck, the diversity ratios move back toward greater equality. The period of extreme male reproductive skew appears to be specifically associated with the social and economic transformations of the pastoral and early Bronze Age periods, rather than being a universal or constant feature of ancient human mating. The burial site populations that ancient DNA has been most informative about include Yamnaya and related steppe pastoralist populations from approximately 5,000 years ago. These were the groups whose westward expansion into Europe has been documented through ancient genomic analysis as one of the most dramatic demographic events in European prehistory, an expansion that replaced much of the existing European genetic landscape in a historically brief period. The reproductive patterns within these populations, as reconstructed from skeletal and genetic evidence, show characteristics consistent with significant male reproductive skew. Patterns that the burial record also reflects through dramatic differences in how male individuals of different apparent status were treated at death. The Yamnaya Kurgan burial tradition, placing high-status males in elaborate earthen mounds with significant grave goods while other individuals received simpler interment, creates a material record that, combined with the genetic evidence, suggests strongly stratified reproductive access. The men in the elaborate kurgans were genetically more likely to be represented in subsequent generations' ancestry than the men in simpler burials, which is consistent with wealth, status, and reproductive access being correlated in these populations in ways that the genetic bottleneck reflects. But the Bronze Age and late Neolithic are not representative of all of ancient human history. The question of what came before, what mating patterns characterized the vast majority of human evolutionary history during the Paleolithic, requires different lines of evidence and produces different answers. Ethnographic data from documented hunter-gatherer societies provides the most direct window into the mating patterns that characterize the lifestyle humans lived for most of evolutionary history. By the way, if you want to go even deeper into the secret lives of ancient humans, I just launched a full course called The Secret Lives of Ancient Humans. 10 modules, 3+ hours of content covering everything we never talk about on YouTube. What they feared, how they communicated, what hunted them, and much more. Plus an exclusive PDF guide included. Link in the description. See you inside. The cross-cultural anthropological record on hunter-gatherer mating is complex and does not resolve to a single universal pattern, but some consistent tendencies emerge from comparison across many documented societies. Pair bonding is present in virtually all documented human societies. The formation of at least nominally exclusive sexual and reproductive partnerships is a human universal in a way that it is not in our closest primate relatives. This near universality of pair bonding, expressed in different cultural forms ranging from formal marriage ceremonies to informal recognized partnerships, suggests that pair bonding is a fundamental feature of human social organization, with deep evolutionary roots rather than a purely cultural invention. However, strict lifelong sexual exclusivity within pair bonds is substantially less universal than pair bonding itself. Ethnographic surveys find significant variation in the cultural rules around pair bond exclusivity, ranging from strict monogamy in some societies to culturally accepted polygyny in many, to various forms of institutionalized or tolerated extra-pair mating in others. The majority of documented human societies, when their formal marriage rules are examined, permit polygyny, one man having multiple female partners simultaneously. Though in practice most marriages within even polygynous societies are functionally monogamous because most men cannot acquire the resources required for multiple wives. The distinction between formal cultural rules and actual behavior is critically important here, and is where most popular discussions of this topic go wrong in either direction. Societies with formal monogamy rules show significant rates of extra-pair paternity, cases where genetic paternity does not match social paternity. Studies measuring extra-pair paternity rates in contemporary and historically documented populations find rates ranging from approximately 1 to 10% of births in most studied populations, with some outlier studies finding higher rates in specific contexts. Societies with formal polygyny rules show the opposite complication. Formal permission for multiple partners does not mean that most individuals actually have multiple partners, since resource constraints limit actual polygyny to a minority of men who can afford the material costs of additional wives. The picture that emerges from ethnographic cross-cultural analysis is of a species whose baseline mating system involves pair bonding as the primary reproductive partnership, combined with significant variation in the exclusivity of those partnerships, and with a minority of high-status or resource-wealthy males in many societies achieving substantially higher reproductive success than average through access to multiple partners. This pattern, predominantly pair-bonded but with meaningful variation, and with higher-status males achieving higher reproductive success in many social contexts, is consistent with the anatomical evidence from body size dimorphism and testis size, which both pointed to a mating system occupying the moderate middle ground of the primate mating system spectrum rather than either extreme. The social organization of Paleolithic hunter-gatherer groups provides additional context for understanding the likely mating patterns of most of human evolutionary history. Hunter-gatherer bands typically consist of 20 to 50 individuals, including multiple family groups with overlapping kinship ties. Within these small groups, social transparency is high. Everyone knows what everyone else is doing. Reproductive partnerships are visible to the entire community, and the social costs of behavior that violates community norms are immediately apparent and directly enforced. In this social context, extreme polygyny, one man monopolizing reproductive access to many women while most men reproduce little, is structurally difficult to maintain. The small group size means the few men being excluded from reproduction know exactly who is responsible for their exclusion, have daily social contact with that individual, and have strong incentives to resist an arrangement that directly reduces their own reproductive success. The social dynamics of small, transparent communities tend to constrain extreme reproductive monopolization by dominant males in ways that the large, anonymous social environments of agricultural civilizations do not. This structural constraint on extreme polygyny in small hunter gatherer bands is part of why the genetic evidence for high male reproductive skew appears specifically in the Bronze Age transition rather than throughout human prehistory. Small-scale hunter-gatherer social organization provided structural barriers to extreme reproductive monopolization that agricultural civilization removed by creating both the wealth basis for supporting multiple wives and the social complexity that made enforcement of reproductive monopolies more feasible. The lifespan context of ancient human mating patterns is another dimension that fundamentally shapes what number of partners means in ancient versus modern contexts. Ancient human lifespans were not as short as popular imagination suggests. The low average life expectancy in ancient populations was primarily driven by high childhood mortality rather than by short adult lifespans, and adults who survived to reproductive age could live into their 40s, 50s, or beyond. However, life expectancy was shorter on average than in modern populations with access to modern medicine, and several specific features of ancient demographic reality shaped mating patterns in ways that have no modern equivalent. Maternal mortality was high throughout most of ancient human history, meaning that many women died during or shortly after childbirth. Paternal mortality from hunting accidents, warfare, predation, and other hazards was also significant. The result was that many ancient adults found themselves widowed, and potentially seeking subsequent partnerships, not through choice or dissatisfaction with an existing partner, but through the demographic reality of losing partners to death at rates far exceeding anything typical in modern developed country populations. Serial monogamy, sequential pair bonds with different partners over a lifetime without overlap, was probably a common pattern in ancient populations precisely because the early death of partners made it a demographic necessity rather than a cultural preference. The average ancient human may have had more lifetime sexual partners not primarily because of promiscuity, but because they outlived partners and formed new partnerships in a world where forming partnerships was a survival and reproductive necessity rather than an optional social arrangement. The distinction between simultaneous multiple partners and sequential partners across a lifetime is crucial for honestly answering the question of how many partners ancient humans had, because these are very different social and biological phenomena despite producing the same number in a simple count. A modern person who has 3 partners over the course of a 40-year adult life after the death of 2 previous partners has a very different relationship with partnership than someone who has 3 simultaneous partners. And the ancient demographic context made the sequential pattern common in ways that say more about mortality rates than about fundamental preferences or mating system characteristics. The genetic legacy of ancient mating patterns is visible in living human populations in ways that researchers continue to refine. Modern human genetic diversity at the population level reflects the combined effects of all the mating patterns that have operated across human evolutionary history. The modest male reproductive skew of Paleolithic band societies, the more extreme skew of Bronze Age pastoral expansions, the various forms of socially organized polygyny and enforced monogamy of different historical civilizations, and the ongoing demographic mixing of modern populations. The specific Bronze Age male bottleneck that ancient DNA analysis has identified, the period where one man per 17 reproductive women appears to have characterized some populations, represents a historically significant departure from what came before and after, and its timing with the expansion of pastoral warrior cultures, the development of heritable wealth, and the emergence of the social hierarchies that concentrated resources in elite male hands provides a compelling narrative of what drives extreme reproductive inequality when ecological and social conditions allow it. The answer to how many partners the average ancient human had is therefore not a single number, but a distribution that varied substantially by time period, social organization, ecological context, individual status within their community, sex, and the demographic accidents of survival and death that shaped partnership opportunities. For most of the Paleolithic, the vast majority of human evolutionary history, the probable pattern was primary pair bonding with meaningful but not extreme variation, constrained by the social transparency and structural limits of small group living, combined with the serial partnership sequences that high partner mortality rates necessitated. The genetic and anatomical evidence suggests that some degree of male reproductive skew existed even in this period. More than strictly equal monogamy, less than the harem polygyny of gorillas, consistent with a pattern where socially dominant or more skilled males achieved somewhat higher reproductive success without monopolizing reproduction to the degree that later social systems permitted. For the Bronze Age transition, the evidence points toward a period of dramatically increased male reproductive skew concentrated in expanding pastoral populations, reflecting the intersection of heritable wealth, warrior social hierarchies, and the removal of the structural constraints on reproductive monopolization that small group living had provided. For the agricultural civilizations that dominate the historical record, the formal diversity of marriage systems, from the polygyny common in many African and Middle Eastern societies to the more enforced monogamy of later European Christian societies, masks the behavioral reality of significant extra-pair mating in virtually all studied populations, combined with the higher reproductive success of elite males that wealth and power enabled. The two skeletons in that European burial site, when their genetic relationship is finally determined and their story is reconstructed from bone and DNA, are not telling us about what ancient human mating should have looked like or what it means for modern people. They are telling us what it actually looked like in that specific time and place. One specific data point in a population that was itself one specific data point in the long, varied, ecologically and socially contingent history of how human beings organized their most intimate relationships across time. The biology set boundaries. The ecology shaped what was possible within those boundaries. The social organization of each specific period and population determined what actually happened within the possible. And the actual answer for any individual ancient human depended on all three of those things simultaneously, which is exactly why there is no single number and why the search for one tells you more about the modern desire for simple answers than about the complex reality of ancient human lives.